today I'm presenting a guide to molecular interactions and how to research and retrieve the data via our database called INTACT. I'm a senior curator in the molecular interactions team. And you can find our contact details on the bottom of the slide. I will show you three different aspects of molecular interactions. I'll start with an introduction, then I will showcase searches using the INTACT website, and finally we will look at the PSICQUIC tool and IMEx collaboration that provide data integration services. So let's start with an introduction to protein-protein interactions and networks. Protein-protein interactions are the physical and selective contacts that happen between pairs of proteins, and these are characterized by happening in particular molecular regions and in a defined biological context. An interactome is the totality of these protein-protein interactions that can happen in a cell, in a whole organism, or any other biological context. And the protein-protein interaction networks are simply the graphical representation of such interactomes, where proteins are represented as nodes and interactions as edges. Why do we look at protein-protein interactions rather than genetic interactions? Because once the DNA is transcribed into RNA and translated into proteins, most of these proteins actually don't have one unique function. They have multiple functions and are found in multiple networks. So by using the protein-protein interactions and building networks from these interactions, we can do two major things. We can identify the function, we can predict the biological function of so far uncharacterized proteins, because proteins that cluster together in a network can be presumed to have similar functions, and this is called Gilbert association. Secondly, we can improve the characterization of protein complexes, pathways, and biological processes using these networks that we build from protein-protein interactions by using these interaction networks as draft maps. In all these cases, further studies are then required to drill down into the exact functions of these proteins or complexes or the details of pathways and processes. The networks can also be used for further exploration of biological functions. So for example, we can overlay data from interactions with metabolites and therefore represent metabolic processes. We can overlay gene regulation and cell signaling information to turn these, by default, undirected edges into directed edges on a network. So let's have a look at some of the characteristics of protein-protein interaction networks. As I just said, they are by default undirected. In big networks, more than 80% of the proteins are connected in one giant cluster. And these networks are characterized by the small world effect and the scale-free aspect of networks. The small world effect means, in the case of biological networks, that any two proteins in the network are on average only separated by 6 steps. And many of these interactions are separated by much fewer steps. So if these networks are so tightly connected, based on the characteristic of the small world, how come that perturbations in a single node don't have dramatic consequences for the network? And that's because of the second characteristic, its scale-free character. Here, most nodes in the scale-free network have only a few connections to other nodes. So these are the white nodes in the diagram on the right, whilst a few hubs or nodes, the gray ones on the right, are connected to many other nodes in the network. This characterization of how many connections a node has is also called the degree of the network, a degree of a node. So if you plot the degree of all the nodes in a network, you will find that the majority of proteins have a very low degree. so the top left on this graph, whilst a few nodes are highly interconnected and therefore have a high degree. And the last characteristic I'd like to highlight is the type of nodes that we find in these networks. So we have two different special types of nodes, and these are proteins that interact, so high degree proteins that interact in certain biological contexts. So some of these nodes interact with many other proteins in the same metabolic situation, the same process, same pathway, the same complex. And these are called party hubs, because all of these proteins together occur at the same time in the same space. So the blue or yellow hubs in this diagram on the right. And then there's some hubs that are actually combining several clusters within the network, and they're called date hubs, where proteins combine different metabolic pathways or processes by being members of these particular different processes. So they can occur at different times, say at different cell cycle points, or in completely different spaces in the cell. So if failures occur at random in these scale-free networks, then the vast majority of proteins are still connected because they have just such a small degree of connectivity. And the likelihood that a protein hub would be affected is small. And even a single hub failure usually doesn't lead to a loss of connectedness of the whole network, because there's so much redundancy in the different connections that this network has. However, if we lose several major hubs from the network, especially several date hubs, the ones that connect different metabolic pathways, then the network gets turned into a set of isolated graphs, and then the network becomes vulnerable to attack. So many, but not all, cancer proteins, for example, are hub proteins. The other thing we need to remember is that whatever you find in a database is just that amount of data, or that part of the data that has been curated. And there's a vast amount of data still out there that we don't capture. And there's also a vast amount of potential data that hasn't even been identified by experimental methods. And this is due to the popularity bias. Interesting or important areas of biology attract more funding, and therefore there are more studies in that field, and therefore there's more data for those particular aspects of biology in the databases. Also, it's been shown that some cancer networks don't behave this way, and you can see more about this in this publication by Schäfer et al. Okay, so let's have a look at the different protein-protein interaction detection methods that we can use. There are literally dozens of different methods, and I've just highlighted a few here. And we generally separate them into two different kinds, high throughput and low throughput methods. High throughput methods in the main covered by the classic yeast-2 hybrids, which are still used very much and now highly automatized, and a huge plethora of affinity purification with mass spec identification methods. And these are usually used for large-scale analyses, interactome level experimentation. And then there are loads of different low throughput experiments. Often these are used to validate experimental findings from large-scale experiments, or drilling down into more functional aspects of these interactions. One example is X-ray diffraction, to really drill down into the details of the interaction of these proteins. One thing we need to remember is that not any single method can accurately reproduce what's happening in the cell, and that all of these experiments are fundamentally artifactual. So if you want to explore the entire interactome of an organism, you cannot rely on one single method, simply because some proteins won't be identified, or some protein interactions won't be identified with certain methods because of the biochemical and biophysical features. And on the other hand, depending on the methods, we can also create a number of false positive interactions. If we, for example, deal with a whole cell extract and you're throwing all the proteins from the cell into one test tube, you will find interactions that are biophysically possible, but biologically would never happen. We characterize these interactions into 4 major types. One are direct interactions between 2 proteins, so using methods that show that these 2 proteins are directly physically interacting with each other, and that's the equivalent of a face-to-face conversation between people. The second aspect are physical associations of co-complexes, where 3 or more participants are found in the experiment. They're also known as NR interactions. And these can be used to characterize larger protein complexes. These are often identified using pull-down or co-amino precipitation methods, but one has to be careful to have a look at the biological context, as I just mentioned, because oftentimes these pull-down methods capture more than one single complex or members from one single complex. An equivalent is a conversation, a group conversation between people where everyone is in the same space and time, but they don't all directly interact with each other. There are a number of methods that identify co-localizations of proteins, also known as associations, where 2 or more participants are in the same space and time, but the methods don't actually provide physical interaction evidence. And these are methods mainly based on confocal microscopy studies and proximity ligation assays, to name 2 major examples. And lastly, there's a type of direct interaction that also gives us information about the function behind this interaction. These assays are almost always conducted in vitro using purified proteins. And these can be enzymatic assays, SPRs, crystallography. They don't have to have just 2 proteins in interaction. They can have more, but the interaction method defines details about their physical interactions more closely. Additionally, we also capture the binding regions where they've been experimentally shown. So for example, there might be deletion constructs that have been used within the experiments, and then we can capture exactly which regions are required for these 2 proteins to bind to each other. Now, the other thing we need to consider is what do we do with these co-complexes that are identified by many, many different interaction experiments. As soon as you have more than 3 proteins in an interaction, there could be all sorts of different conformations in reality. And you don't know exactly which protein interacts with which one unless the experimental detection method actually showed you that. And the only one that really drills down into that is X-ray crystallography and electron microscopy. All other methods will not be able to tell you exactly what the binary interactions within these co-complexes are. So we have 2 options. We can either assume all proteins interact with each other. That's the matrix expansion. And you can imagine as the interactions become larger and you have dozens or hundreds of interactors, the number of false positive interactions we create by matrix expanding these is vast. So most protein-protein interaction databases, and that includes INTAC, use the spoke expansion model, where one protein is assumed to interact with all other proteins in the experiment. Where the experimental method has an experimental bait, so a pull down or a co-IP experiment, then this bait becomes the center of the spoke in the expansion. Otherwise, there's a complicated mechanism to identify the spoke. This obviously loses some true positives, but it really reduces the false positive interactions and also keeps the databases manageable compared to the exponential expansion you would find in matrix expansions of large interactions with many interactors. Okay, so this is the introduction. Now we have a look at the characteristics of the intact database, or the specifics of the intact database. Intact data and content is freely available. We now have over 1 million binary interactions, so that includes the bespoke expanded NREs. They're taken from over 22,000 publications of experimental data and represent over 120,000 unique interacting molecules. Because apart from proteins, we also capture interactions with small molecules or nucleic acids. We are a member of the IMEx consortium, which I will mention again later in this webinar. Now here you need to bear with me. How do we access Intact? Well, normally you would access Intact through the first URL here, www.ebi.ac.uk/Intact. However, we are currently deploying a brand new website, which is under development and can be found on our beta site, which is www.dev.ebi.ac.uk/intact/beta/hold. What I'm presenting today is what is on the second website, plus some features that are about to be deployed in the next few days. So if you're having a look at this beta website, and maybe Kalpana can put the link into the chat for you, you won't see all the features I'm presenting. If you're watching this webinar on recording, then hopefully all the features I'm presenting today will be available. And if you're watching this webinar in the future, in a few months or years' time, then everything should be back on our normal website, the ebi.ac.uk/Intact website. So I apologize for this in between, but we really want to showcase our new capabilities of the new website. In any case, you can always download the data in our standard formats from the FTP side, or use the PSICQUIC web service, which I will come to in the last section. And if you really want to, you can download the whole thing, the data and the software, and install your own local Intact node. So where does our data come from? We curate either already published data from publications, or we take direct submissions. We really like direct submissions because then we can make sure we capture all the data in its depth and detail that we can. And we can also provide feedback to the authors about the consistency of the data and if we have captured everything that they actually have available. So oftentimes there's more data, more features of these proteins available that does not get published, not even in the supplementary material. And we can capture all of that during the direct submission process. And we keep the data confidential until the publication itself is out. In both cases, we can deal with large-scale, high-throughput interaction projects, and we have a bespoke semi-automatic pipeline to deal with essentially Excel-type data input that is then converted into our XML format to go into the database. And we work with our submitters on this pipeline. We have a very detailed curation schema. As I mentioned, everything starts with a publication, whether it's already published or a pre-publication manuscript. And then we capture every experiment and every interaction from these experiments. There are some protein-protein interaction databases that will only capture the interactions that are relevant for their domain specificity, but we capture everything that's found in this given paper. Then obviously we list all the participants per interaction. And what is special for us and our IMEx curation partners is that we drill down one level further and we actually describe the features of these participants. And these features can include the binding regions that are required for 2 or more interactors to bind, the mutations, which I'll come to in a moment in more detail, kinetic parameters, where we're dealing with enzymatic assays, and simple construct tags like GFP or HA tags that are used for the protein purifications or identification as part of the experiments. Curators use a very detailed curation manual and CVs, that stands for controlled vocabularies, to make sure every curator curates to the same rules and that the data is standardized. Every entry into the database is checked by a second curator for accuracy and completeness of the curation of the particular paper. And the second curator can reject the entry, and then the first curator needs to make changes. When we deal with brand new data types, this often means a larger discussion between curators within the consortium, and often the creation of new CV terms, so we can really accurately capture new types of interaction data. And then the data gets released roughly every 2 months. We also tag a number of our publications with specific data sets. We call this contextual curation, and I would like to highlight 3 data sets we're particularly working on at the moment. The first one is the coronavirus data set. That is a collaboration of all IMEx partners that was started in 2020 when the pandemic started. And a number of curators focused on curating interactions between coronaviruses and their hosts, and also viral-viral interactions. This is not limited to SARS-CoV-2. This is broadly for all coronaviruses, so that orthology experiments can also be conducted and orthology comparisons. And the other 2 data sets are linked to each other. We have a strong focus on curating interactions that are affected by mutations, and these can be specifically captured, which I will show you examples of later. And within this data set, we have a curator who's focusing on variants that are linked to rare diseases that affect interactions. Okay. So now I will show you some examples of how you can search in INTACT. And because this is a mixture of capabilities of the 2 instances we have live at the moment, I'm presenting these as screenshots. Our front page is very simple now. You have some quick entry points through the tiles if you want to have a look at data sets or submit your data. On the right-hand side, you have some details of how you can stay in touch with us, but we're focusing mainly on simple searches today. Before we go to that, I will highlight the interactomes that we are creating. This is brand new. We didn't have this previously. So we pre-compute the 16 most popular and populous interactomes directly. And if you click on the species icon, you will get the results for all the interactions with proteins from these species. Or if you click on the button below, the purple button, you can directly download the data for this species in XML. So the first search example I will give is searching for CDK1, cyclin-dependent kinase 1. I've chosen this example because it's a ubiquitous protein, but it also throws up a few interesting characteristics of searching with a gene symbol. We provide you with suggestions when you start typing in the search box. And as you can see, not all of these suggestions include CDK1 as a gene symbol, but they're all called, if they're not called CDK1, they're called cyclin-dependent kinase 1 as the protein description. And there are 65 potential interactors that have the string CDK1 associated with them, either in the name or as a synonym or alias. Curiously, the human CDK1 is only found in the second page, so sometimes you have to start scrolling for what you're looking for. So in this case, I'm just hitting enter, or you can click on the search icon, and then you will get a list of all the interactions with proteins that are somehow identified as CDK1. There are nearly 2,000 of these. And if you have a look at the table, I'm not sure how well you can see it. There's lots of information in the table. There are the symbols and identifiers for each interactor, as well as what type they are. So in these first few interactions, they're all proteins that are linked to UniProt. They're all human proteins, but the experiments have been done in different experimental hosts. So for example, in yeast or in vitro. It shows you the interaction detection method. There's always a link to the publications. Then what type of interaction, which I mentioned earlier, if they're physical or direct or just associations, and many other links. And we'll come to some of these details in a moment. This is a very long table. It has over 40 columns. So if this is too much, then, or potentially over 40 columns, you can then untick some of these columns and make the table display specifically the columns that are of interest to you. You can also display the list of interactors. So in this case, the nearly 2,000 interactions are included, are made up of over 1,000 different interactors. These are mostly proteins, but there will also be other types of interactors when you scroll through this list. And as you saw, there's this error message up here, because we can't display a network view for more than 1,500 interactors because it's too big to display, too computationally extensive, and also not very useful for the human eye. So you have various options. You can use the Intact app for Cytoscape, which I will mention later again. You can make a new search with a more specific search term, or you can use the filters, which is the first thing I will demonstrate now. So we've implemented a number of different filters, and in the first instance, we will look at the species filter. Here you can see that you can select different species, multiple species, and you can also select if you want interactions with proteins that are just from that species. So intra-species interactions, which is the right-hand column, and you toggle the little button on the top right, or you want to have all interactions from a specific species, but you don't mind if these interactions also contain proteins from other species or other types of interactors. So for the purpose of the demonstration, I've chosen mouse and mouse with other interactors. So here you have a network that has lots of separate clusters. The main interactor here is the mouse CDK1, and if you hover with your mouse over the nodes, you get this little pop-up information box. But there are also many other interactors that came up with the search term CDK1. For example, CDK10, there's also CDK19 and 17, and all sorts of others. So you can see a search for a gene symbol provides you with a large overview, but potentially not the detail that you would expect from this search, because it is just a search string. So what can we do? For example, we can use the UniProt identifier. This one is the UniProt identifier for CDK1 in human. And you can see even that might not be unambiguous, because CDK1 in human has got several isoforms, and we have curated one interaction each to isoform 1 and 2. We only annotate interactions to isoforms or post-process chains in other cases if the experiment has shown that this interaction is unique to that particular isoform, at least in the context that the experiment was conducted. So we can either choose to just search with the canonical identifier, and then we get this central wagon wheel where every interactor interacts with our search, original search term, CDK1 in human. We can also, in the search box, just hit enter, and then we capture the two interactions as isoforms 2. So here we have the isoform 1 that's actually connected to the main wagon wheel through a common interactor, and the isoform 2 just interacts with catnal 2. Okay. So the other thing to highlight here is the visualization of the network. As you can see, the nodes have got different colors, and these are associated with the species. Most of these are light blue for mouse, but there's some interactions also with human, with Drosophila, and with C. elegans, as well as some viral proteins. And that is not surprising. Oftentimes you get interactions with different species, either because the construct, due to the availability of constructs. So, for example, the sequence between mouse and human might be almost or directly identical. And if the construct for the other species was available, then the researchers will use that, and we will capture exactly what was done in the experiment. So we don't make orthology inferences to the same species. We just use what's been shown in the paper. And the interactions, for example, with C. elegans or Drosophila stem from functional assays where the ubiquitous function of this kinase was investigated using various interacting partners from orthologous species. Finally, you can see those brown triangles. Those are identifying chemicals that are part of interactions. Yes, they're chemical molecules, small molecules. And the edges, they're not so easily visible in this bigger diagram, but you will see more examples later. They identify the top-level types of interactions again. So are there direct binaries? Are there physical interactions? In this case, we also have some phosphorylation interactions highlighted in yellow because we're dealing with a kinase. And those interactions that are dotted come from those spoke expansions. So these are interactions that may not be direct, that come from experiments where these two proteins and other proteins are included. We can collapse these edges by unticking the expand button on the left, and then we get a different bit of information on the edges. And this is information about the amount and depth of confidence we have in these edges, in these interactions between two proteins. The thickness of the edge represents how many evidences we have in the database, and the color is correlated with the MI score, the molecular interaction score. This is a score that takes into account the number of interactions we've curated, the number of publications these interactions come from, and the type of interaction. So are these high-level biophysical, for example, crystallography experiments, or are these just loose co-localizations? And this score is calculated and then indexed. And so the actual score is in a range of 0 to 1. You can adjust this score, you can adjust the network by changing the range of score that you're displaying. So you can use one of the other filters on the top. And then, for example, you can set the minimum to 0.45. That removes all interactions from the view and from the table that contain only one evidence. So now what is left in this particular view are all interactions that have at least 2 evidences for this interaction. And if we drill down into some of the details, if you click on the edge, it will show you a summary in the tooltip and then display the interactions that are behind this edge in the table. So in this case, there are two interactions, a co-IP, an anti-tag and an anti-bait co-IP, for example. And finally, on the left-hand side, you can also click on interactions affected by mutations. So we highlight here in pink all the interactions and the interactors that are affected by mutating a particular amino acid in the sequence. And again, by clicking on an edge, you can get more information. And let's have a look at the details of this particular interaction. So we can click onto this file symbol on the left-hand side, and that opens up the interaction details view. And the interaction details view on the bottom repeats most of the information we already saw in the table. And then we have the view, the complex viewer, so-called, that was developed by the Rapserver lab in collaboration with us. And that highlights the features on the actual sequence of the proteins. So in this case, you see the mutation from threonine to valine in position 37 of CDC6, as well as the GFP tag that was used. Many of these terms already in the results table are hyperlinked to the relevant ontologies that describe these terms. So if you're not sure what an anti-tag co-IP is, you simply click on the link and it opens up all the details about this term in the ontology. You can toggle through these tabs in the table similar to the results page, and you get details about the participants. So again, you have links to the UniProt details for the two proteins. And if you click on the features, you can get the details of this mutation, for example. So here it tells you that this mutation disrupts the interaction. So if you mutate CDC6 position 37 to valine, then this interaction does no longer happen. Finally, there are other options of displaying features in the complex viewer that you can have a look at in your own time. Okay, let's have a look at a couple of other types of filters we implemented. Here, my search term was a kinase, and I'm interested in just having a look at the phosphorylation interactions, phosphorylation assays that result in interactions with CDK1. So I can filter this out, and there are now 25 interactions left. And finally, we've spoken a lot about the spoke expansion of co-complex experimental evidences. If for your particular study, you don't want to trust these expanded binaries by spoke expansion because of the potential of false positives, you can filter these out, and then you're just left with binary interactions that come from experiments that show that these two proteins are definitely physically interacting with each other. So you just toggle the button in the expansion method. Okay, so that's basically it about simple searches. Now, if you want to search for more than one interaction term, interactor, you have the option of the batch search. You can simply type a list of identifiers into the box, and this can be either comma separated or in separate lines. You hit the next step button, and then the identifier resolver will tell you what options there are for this search term. Now, if you use UniProt identifiers, these are already unique, unambiguous terms. So for those three identifiers, I only get 3 options, so 1 per identifier. And then, for example, I could say, yes, I want to search for the interactions of all 3 of these with any other interactors. So I hit on search and I get my network view again. This is a highly connected network because the 3 proteins I've chosen are part of a larger complex. So they have a lot of common interactors, both within the complex, but also within the larger biological process this complex is involved in. Now, I could have also searched with gene symbols. These are two gene symbols of proteins from the same complex again. But as we've seen before, gene symbols are ambiguous, and the identifier resolver then shows you the options you have. So here you can then select or unselect the identifiers with which you want to carry on and actually build your network. So I've chosen to use the two mouse proteins again for TERF1 and TERF2. I will have unselected them all and just selected the two mouse ones and build a, in this case, quite small network that contains both TERF1 and TERF2 and all of its interactors. And you can see a few of those are interactors of both of them. So this is a combined interconnected network. This network also shows you a different interactor type with this little kind of inverted triangle that identifies a nucleic acid that's part of the interaction. And finally, you can do a batch search using your own files. So you can create a file of identifiers and upload it, and then search for it. Right, once you've done your searches and you're happy with the results you have, obviously you want to take the results away with you. And you can do this in various forms. You can download the actual network that you've created in PNG or GraphML format. And you can download the table in our standard formats, the MyTab, tab delimited format, the XML, or the MIJSON. I briefly mentioned it earlier. There's an alternative approach of doing protein-protein interaction searches in Intact, and that's through our Cytoscape app, of which the link is on the bottom of this page. I won't have time to to show you more about this. This is for you to explore. The searches are very, very similar to the website, and then you have the added benefit of all the power of Cytoscape to run full network analyses of the networks you build with the Intact search. Now, finally, I will spend a few minutes explaining to you what else you can do and how Intact is integrated in the community of protein-protein interaction databases. So there are many different databases that deal with protein-protein interactions, and it would be great if everyone had started with one unique database. But that's not the case. A lot of these databases were created as domain-specific databases when the sort of larger data generation started some 20 years ago. So we ended up with lots of databases appearing, struggling to be maintained. A lot of them weren't easy to find. And because they're all set up separately, they use different formats, controlled vocabularies, ontologies, and minimum information guidelines, which meant the user had to go to each individual database, do a search, work out how to work the database in the first place, and then download the data and integrate it on their own, on the user side. That led to inconsistency because trying to integrate various different formats is very hard. And obviously redundancy, because the databases weren't talking to each other about which papers they were curating, not in the early days. So we found a compromise where we said, well, the different database instances still have to remain for all sorts of reasons. One of them is that the domain-specific databases want to serve their users their domain-specific information. But what we can do is we can provide users who want a global overview with a unique singular search interface. For that, we had to convince these databases to change their formats, controlled vocabularies, ontologies, minimum information guidelines to one that's standard for the community. So the data became consistent And in one of the two examples I'll show you, the data is also now non-redundant. So the first initiative was the Proteomic Standards Initiative for Molecular Interactions, also known as PSIMI. And the PSIMI initiative created the community standards and controlled vocabularies that we're using to this day. And it created PSICQUIC, the Proteomic Standards Initiative Common Query Interface. And through this Common Query Interface, we can also give you the PSI score, which is the equivalent of the MI score, but across all services in PSICQUIC. And secondly, 12 databases came together to set up the International Molecular Exchange Consortium, IMEx. These IMEx partners all use the PSI standards, but they also collaborate on the curation side. And we're now all curating into the same environment, which happens to be the INTACT environment. And this produces a non-redundant data set. So any searches done through the IMEx website or the PSICQUIC service for IMEx, and I'll show it to you in the next slide, are non-redundant results. So the added benefits are that we have a standard format. The default format is our XML. We also have a tab-delimited format, the MITAB. There's now a format for capturing causal statements, which is integrated into MITAB 2.8. And there's an industry of so-called composite databases or data warehouses that combine all this data that is in PSI-MI format. There are also bioconductor packages and Cytoscape apps that make use of these common community standards. So PSICQUIC looks very similar to INTACT. You do searches in the same manner by using identifiers, and there are nearly 30 members or 30 services of PSICQUIC. PSICQUIC is hosted by the EBI, but the individual service providers are responsible for their service. So if a service is down, like in this snapshot, there were quite a few that were down, that is the responsibility of those service providers. When you do a search, this is again the human CDK1 identifier, you then get into the results for each individual PSICQUIC service. And if you click on one, for example, INTACT, it opens a table. And from there, again, you can download the data in our standard formats of XML and MITAB. If you have fewer than 5,000 interactions in your search results, or you unselect some of the databases, you can cluster this query. And so you get a single line for each unique interaction between two proteins, with the interaction evidence is concatenated in further fields. If you want to find out the characteristics of each of these PSICQUIC services, you can click onto the little magnifying glass, and that brings up this pop-up tooltip. You can get further information from the PSICQUIC registry. That includes the number of interactions that are available from each database, the search URLs, and again, the tags of what characterizes this database. And here I'm comparing the INTACT and IMEx characteristics to Interporc. So you can see there's hardly any difference between INTACT and IMEx, except INTACT has got the tag imported because members of the IMEx consortium have imported their data, or we have imported the data from members of the IMEx consortium when we decided to build, to use INTACT as the common curation and development platform. But everything in INTACT as well as IMEx is still evidence-based and manually curated. In comparison to that, Interporc is a data warehouse. It focuses just on protein-protein interactions, whilst INTACT and IMEx also capture interaction of small molecules and nucleic acids. And it includes interactions from prediction algorithms. And a lot of the warehouses do this. They bring the predicted data in, which can be very useful, especially for species that are underrepresented in certain aspects of biology. Here's the link to the IMEx website. So you can do exactly the same searches through IMEx, and then it will provide you with the non-redundant, manually curated data set. And on the bottom of the slide here, you see the 12 IMEx partners plus the 2 observers. So in summary, curated interactions in a database only represent a fraction of the published knowledge, and even less so compared to everything we don't know yet. Different databases have different curation strategies and depths. Text mining can actually increase the number of PPIs in a database, but you're lacking that quality control of manual curation and checking process that I explained earlier. And we always have to be mindful that everything we do in a Petri dish, essentially, may not be the same to what actually happens in the cell. So every result, every network analysis needs to be taken with a pinch of salt and then analyzed in against the biological background for which you yourself will be the expert. And finally, curators are humans and we make mistakes. So if something really doesn't make sense to you, then please get in touch and we'll have a look, because sometimes little mistakes still happen despite the checking process. Okay, so how do you get in touch? There's a feedback button on the top. We really, really appreciate feedback on our new website or any questions you have. If you have data to submit to us, please submit it through this the quick access link on the front page. You can follow us on Twitter, and you can also sign up to our newsletter. We don't send many newsletters, but in the next few months, there might be a few more to keep you updated with the development of our new website. And with that, I'll leave you with a few helpful links that you can look at in your own time. There are also many other tutorials on the EBI training website related to Intact and our sister database Complex portal. And with that, I'm thanking the team leader Henning and coordinator Kalpana, my fellow curators Margaret and Sandra, and all of our IMEx curators, and our developers Noe, Anjali, and Elliot, as well as our funders, EMBL-EBI, Open Targets, Wellcome Trust, BBSSC, and the NIH. Thank you. I hope we have a bit of time for questions, and I will put my camera back on and take questions. So maybe Kalpana, you can read them out for me, what you haven't answered yet, because I haven't paid any attention to chat.
Yeah, so interestingly, we had a lot of questions today. So currently I'm trying to answer is there API for Intact available? So if you can answer that, I'm taking care of the rest of the questions.
Yeah, so at the moment, the nearest we have to an API is the PSICQUIC service. You can integrate that into scripts and ping it directly. I know that with the new website, eventually there will be a new web service as well. That hasn't gone live yet. So if you sign up to a newsletter and follow us on Twitter, then yes, we will let you know as soon as the API is available.
And so we have other question. So interactomes in Intact are available only for few species. What about interactomes of trypanosomes like Leishmania?
You can build them yourself. So you can make the search and search just for the species of interest and build your own interactome.
So there's another question from Jyoti. I think she left the webinar, but questions are like, I'm just wondering, how does the interactome clusters, like what are these categories? Is it 4 classes of interactions we just finished talking about? Well, I mean, I couldn't get more context on this question. I'm still, I think it is still not exactly defined. But I think she's, yeah, I think she is talking, I think she might be describing what are the clusters, like cell signaling type of in the networks that we are described in the initial part of the webinar. Maybe that, but I'm not sure about. Okay, so is it possible to submit more than one protein to Intact? Well, I have no, I mean, this question is again, I mean, is it for querying or building the network? I mean,
looking at the timestamp, I think the question was asked before I showed the batch search. So you use the batch search to search with more than one protein.
Okay, great. Why does the graph look often completely different when I try to reproduce it later at a later time?
That's because we add data. So each release produces new data, so the graph might look different. So this is a really good question, actually. And it highlights how important it is that you capture the version of the database instance that you're using for your analysis. So always take the version or the timestamp at the point where you download your data.
So for quantitative proteomics study, where we aim to identify differentially expressed proteins, one should go for PPI if affinity proteomics not done?
Oh, yeah, that's quite an in-depth question. I think it depends on the context. So you can always have a look what data is already out there.
Yeah, but we do not consider the expression level for our interaction to be added in the network, right? And for intact curation, we do not bother about the level of expression. We just wonder about how far the interaction is being detected and how solid is the strength of the interaction. So yeah. This is one more level which is not actually intact model yet.
Yeah, we don't have expression data, but I don't think the question is about expression data in our database. But if there's no expression data available yet, yes, you can start with protein-protein interaction data, yes. But it won't tell you if these are necessarily expressed at the same time because of the fundamental artifactual characteristic of the experiments. So I wouldn't extrapolate from PPIs to co-expression.
Okay, so I was looking for this gene which codes for protein gene RD29A in Arabidopsis thaliana, but I'm not able to find it here in the database. What could be the reason?
Well, we might not have any interactions
reported. Yeah, reported for it.
Yeah.
So, or if maybe there is, once we have such publication and then we'll definitely have this answer.